Hi, Assalamualaikum. My name is Muhammad Afiq bin Hussein. I'm from Kuliah 3 and I'll be talking about the characteristic of bacteria. In the characteristic of bacteria, there are three components that I'll be talking about. Firstly is the types of bacteria, secondly is the shape of bacteria, and thirdly is the growth arrangement of bacteria. So moving on to the types of bacteria. There are two types of bacteria based on the cell wall. So the first type of bacteria is that the gram-positive, and the second type is that the gram-negative. So let's look the differences between both of them. Firstly, both of them have inner plasma membrane. We look over here, inner plasma membrane, and here inner plasma membrane. And secondly, the gram-positive have thick peptidoglycan over here, and the gram-negative have thin peptidoglycan. So remember, the gram-positive have thick. We can look on the diagram. It shows that the three layers. So the gram-positive have thick peptidoglycan or cell wall, and gram-negative have one layer. So we say that the gram-negative have thin peptidoglycan or cell wall. So and then we look, the gram-negative have second plasma membrane, while the gram-positive have no second plasma membrane. Because of this is the second, and we see there is no such thing as the second plasma membrane on the gram-positive, because of the absence of the outer membrane. The gram-positive is easily treatable with antibiotic, while the gram-negative, because they have the second plasma membrane, so they will harder to treat with the antibiotic. And lastly is that the gram-positive appear as dark purple after gram stain process, while the gram-negative appear red after the gram stain process. The reason is that the gram-positive have thick peptidoglycan, so that it can retain the purple color, while when the gram-negative have thin, so they can't retain the color, so the purple color get washed out when we use alcohol. So after we put the safranin, so both of them will have the red dye, but the gram-positive have the darker purple, so it will mask off the red color, while in the gram-negative they don't have the purple color, the purple dye, so only the red color of safranin will color the gram-negative bacteria. So I hope you're clear about that, and we move on to the shape of bacteria. The first shape is that the spirili in plural or spirillum singular. So here the spirillum describe about the spiral shape or helical shape. And second shape is that the bacillus in singular or bacilli in plural. The bacilli describe about the rod shape structure, just like this. It looks like a rod, so nothing to really to say. So the third shape is that the coccus in singular or cocci or cocci, whatever you want to call. So basically it describe about the round or spherical, just like this. And normally these bacteria don't grow as a cell, they grow in a chain, in clusters or in pairs. So basically the bacteria can be characterized in the growth arrangement also. The first growth arrangement is that the staff arrangement. The word staff describe about the clusters of cell or grape-like cluster of cell, just like this. They group in, like many of them group in one place like this, so we call it as the cluster. So over here we look, this means the cluster, and this coccus, which describe the round shape. So we could say that the cluster of round shaped bacteria, Staphylococcus, the cluster of round shaped bacteria. And then moving on to the second growth arrangement is that the strap arrangement. Strap describe chain. So we look the strap arrangement and the chain and coccus, the round, the chain of round, just like this. And moving on to the third arrangement is that the diplo arrangement, which describe about pairs. Look over here, got double coccus or bacteria. Here, over here also. So diplococcus, same with the top. Diplo, which tell about the pairs, and coccus, the round shaped bacteria. So we got the pairs of coccus. Yeah, I hope it is clear about the characteristic of bacteria. So we will move on to the next person.
Hi, my name is Shazmin Adam bin Sharful Azri with the metric number AS12513. And I'm here to talk about bacterial metabolism. Most bacteria are aerobic, and like animals, they require a constant supply of oxygen to carry out cellular respiration. Other bacteria, called facultative anaerobes, are able to grow in either the presence or absence of gaseous oxygen. Some bacteria are obligate anaerobes, which are unable to grow in the presence of free oxygen. Photoautotrophs are photosynthetic, which means they use solar energy to reduce carbon dioxide to organic compounds. There are two types of photoautotrophic bacteria, those that perform anoxygenic photosynthesis and oxygenic photosynthesis. The difference between anoxygenic photosynthesis and oxygenic photosynthesis is that anoxygenic photosynthesis does not produce oxygen gas, while oxygenic photosynthesis produces oxygen gas. Anoxygenic photosynthesis occurs in photosystem 1 only, while oxygenic photosynthesis occurs in photosystem 1 and 2. And anoxygenic photosynthesis requires a unique type of chlorophyll called bacteriochlorophyll. Green sulfur bacteria and some purple bacteria carry out anoxygenic photosynthesis, and they usually live in anaerobic conditions such as the muddy bottom of a marsh. Bacteria called chemotrophs carry out chemosynthesis, which means they oxidize inorganic compounds to obtain energy to reduce CO2. And they also keep nitrogen cycling through ecosystems and live in environments such as deep sea vents 2.5 km below sea level. Move on with heterotrophic bacteria. There's chemotrophs, which obtain carbon energy in the form of organic nutrients produced by other living organisms. For example, parasitic bacteria feed on the tissues and fluids of their living hosts. There's also Cephalotrophs. They serve as decomposers that break down organic matters from dead organisms, which plays a critical role in recycling matter and making inorganic molecules available for photosynthesizers. Bacteria also forms symbiotic relationships in which two different species live together in an intimate way. One of those is mutualism, in which both species benefit from the association. For example, mutualistic bacteria in human intestines, root nodules of soybean, clover, and alfalfa plants. There's also commensalism, in which only one species benefits while the other is unaffected. For example, a bacteria uses up oxygen in the intestines, allowing obligate anaerobes to live. And then there's also parasitism, in which one species benefits while harming the other. For example, the bacteria can be injected deep into damaged tissues and produce toxin when someone stepped on a rusty nail, which can cause tetanus. So we will continue with Hurin.
Hi, and Assalamualaikum warahmatullahi wabarakatuh. My name is Surina binti Razum An-Nazri with the matrix number AS13173. The bacteria diseases in human can be classified into different categories depending on where it is affected. If it is sexually transmitted, for example, Treponema pallidum that causes syphilis, Chlamydia trachomatis that causes chlamydia, and also Neisseria gonorrhea that causes gonorrhea. And another type of bacterial diseases are respiratory diseases such as strep throat, scarlet fever, tuberculosis, pneumonia, Legionnaires diseases, and many more. The most common causes of bacterial lung infections are Streptococcus, pneumonia, Haemophilus species, Staphylococcus aureus, and Mycobacterium tuberculosis. So how do we overcome this? Well, the scientists have found the treatment for this type of diseases, which is the bacterial diseases, which is antibiotics. There are two groups of the antibiotics, a bactericidal antibiotic and also a bacteriostatic antibiotic. What is the difference between the two? A bactericidal antibiotic is the one that kills the bacteria without the reliance on the patient's immune system to help. A bacteriostatic antibiotic is the one that prevents the growth or the organism from multiplying, but it is the patient's immune system which kills off the bacteria and leads to recovery of infection. So we need to take antibiotics accordingly with what we prescribe, because antibiotic will be effective only and only if they reach the minimal inhibitory capacity. As for that, that's why we need to take antibiotic as per prescribed by the doctor. So we go to Sean.
Hi guys, my name is Sean Ashley. My matrix number is 12935. Today I'm going to talk about cyanobacteria. Cyanobacteria are gram-negative bacteria and also classified as prokaryotes. They ranging from 1 to 50 micrometer in width. Cyanobacteria were called blue-green algae because most of them are found in blue-green color. They can perform photosynthesis, which means they also have pigments. The pigments can include chlorophyll A, carotenoid, and phycobilins, which cause them appear in blue-green colors. Cyanobacteria can have other pigments that match the color of chlorophyll, so that's why they can appear red, yellow, brown rather than blue-green colors. Some of cyanobacteria can perform nitrogen fixation because they possess heterocysts, which are thick-walled cells without nuclei. They can serve as food for heterotrophs in ecosystems, such as provide nutrients to fungus by forming lichen. Cyanobacteria can be found in all types of environment, such as fresh and marine, water, soil, and on moist surfaces, harsh habitats such as hot spring, and so on.
So over here we have our bacterial model, and we look. Number one is the cell wall. The function is to maintain the shape of cell from the trigger pressure, and we have nucleoid over here, the regions of circular DNA, and number three, the ribosome, and number four is the pilus, the tubular structure to pass DNA from one cell to another cell, and plasmid, the extracellular DNA, and the number 6 is the inner membrane. This number 6, inner membrane over here, the blue color. And number 7 we have the outer membrane. As you guys can see, the number 7 over there. And lastly we have the flagellum, number 8.